Thank you for the kind introduction. I'm guessing. Hopefully next time I come, my Russian will be better to understand. So I have actually quite a long presentation today, so please bear with me. I'll be talking about some technologies that we have been developing over the years on acoustics and magnetics. And just to give you an idea about what we do, we use technologies, mainly develop and create technologies over the years to apply to different areas. And some of the different areas that we have been interested historically has been bioprinting, tissue engineering, as well as looking into some of the diagnostic areas and applications in cancer tissue systems, which I will a little bit get into today to show you what we can do with these bioprinting tools and how we can apply it to different things. So half of my presentation will be on different acoustic levitation technologies, and I'll gear towards magnetic levitation technologies to build systems that pull together either microgels, spheroids, tissue spheroids, and using self-assembly to create additive biomanufacturing technologies. And one of the directions that we have taken has been always to self-assemble these micro-constructs that we try to first engineer to make functional and then bring them into more complex and larger constructs in a way that we could build complexity. And the top-down approach has been traditionally, which is I think I would say the earlier approach, and still is applicable to many areas in the clinic even, is just making scaffolds and then applying them to and loading them with cells and trying to create functional systems. So I'll mainly talk about how we look at bioprinting from an assembly perspective. And over the years, people started thinking about self-assembly, especially in nanoscale. Self-assembly is what dominates in cells, in microscale. But when you start looking at spheroids, like micro-scale objects, then assembly dynamics change. So the self-assembly becomes harder if you are going to try to control patterns. And therefore, we have been thinking over the years, how can we apply forces, how can we apply methods to actually control how we assemble things. And that's kind of what has created a generation of different approaches. And one of the approaches has been, I'll show you a quick video about how we use robotics and micro-robotics to make constructs. We have looked at using photolithography, which we have borrowed from tissue engineering, to apply to different areas. And we have looked at levitation technologies, which I will mostly cover these areas today. And the standard bioprinting was where we started mainly. So I'll mainly try to go around and discuss on these three areas of technologies, but just to give you some background, we usually make ourselves by building these masks and photocross-linkable hydrogels to make these micro units of tissue constructs. And spheroids also fit into this description because spheroids are also, in a sense, micro-constructs, and if you culture them long enough, as the earlier speakers discussed, they can become tissue units, which you can then assemble with bioprinting, like 3D Bio does, and or you can maybe do systems that you can, again, build more complex architectures. And our initial thinking was if we could use these multiple masks to build different layers of constructs in 3D, but there is a limit to it because after some point, as you build higher and higher, your control over the geometry is not as accurate as you would like it to be. So we really need higher throughput systems to assemble things. And if you look at a single microgel, you end up seeing, for instance, with primary neurons in it, and they are about a millimeter by millimeter with 150 micron height. That's a construct, and if you kind of look at here with the confocal, you see that the neurites are extending to form and travel in 3D, making it more like a construct that could mimic native tissue conditions. Through a collaboration with Dr. Sitti, who was at the time at Carnegie Mellon, we started thinking about using magnets, magnetic 8-axis forces that would move these hydrogels that could have, you can think of these colors as different codings for gels, and move it around so that we could make different shapes and constructs and control everything at one gel at a time. As you would imagine, this is a very low throughput. We can even play Tetris with it, with hydrogels. So that was a neat experiment showing controllable gels, but scalability and automation of this process could be interesting for maybe some in situ type applications, which in the future could be explored. And you can pretty much control this like a stick of your computer or automated with integrated with image recognition algorithms to control things. This is like micro robotics in action for building things. So the main focus I'll spend time on today is acoustics and how we use acoustics to assemble spheroids and cells both in 2D, 3D, and in Z dimension. When I gave a talk here a while back, there was a question from the audience about assembling acoustics in the Z direction, and I told we were working on it, so I'll show you some of those results as well, how we can make multiple layers by standing waves in the Z direction. But first to talk about Faraday and Faraday waves. These are really neat waves that you see if you leave your cup of coffee on the airplane where the waves are traveling between the fluid and air interface. These are Faraday waves, and you can make really a lot of beautiful shapes and structures with these waves by changing two things: the amplitude and the frequency of shaking. Think about holding a glass filled with water like this, and you're just doing this pretty much, going up and down, and it generates these vibrations that lead to all these very controllable, predetermined, you can kind of pre-enter the shape that you want to get your beads, cells, or spheroids to assemble, and they will always take that shape because that's where the resonance is, which I'll show you later. So for instance, in a square well, if you can generate, for instance, these type of standing waves where you know the nodes and antinodes, that allows you to know where the swimming beads or cells would go. So you can pretty much design, you can think about what the forces will be, and then you can translate it to real action. So you exactly know how you assemble these things. So there are two major forces here. One is the acoustic forces that move things, and the other one is once the cells or spheroids are close enough to capillary forces that kind of hold them together. And that's something that we had published earlier. And as you change the shape, like we tried heart shapes, we tried circles, as you change the shape of the reservoir as well, you're actually able to form very complex shapes. So as you shake the surface, what's happening is you're creating these standing waves. That's quite simple. And this is a very high-speed camera showing how things are assembling. Now we stop and change the frequency, and then you will see a cross kind of getting assembled with this system. So it's a quite simple system that just shakes things. Over the years, our thinking has advanced in a sense that we don't only assemble swimming objects, because then you would have to every time make sure that our fluid density is lower so that we could levitate the cells to swim. We now know that by creating again Faraday waves, we could assemble things at the bottom. So you could pretty much put your cell media and assemble things by leveraging the resonance on the surface, which creates these microflows that sweep things together. So you can create a few layers of cells or spheroids. The system that we have built, this is a picture of our system from the lab, is a power amplifier that is a part of pretty much any acoustic resonator, a Z-dimension system. And we used to have this different types of valves and fluidics that we create on top to have the shaking properly so that everything would be assembled here. Over the past year or so, we have been able to actually integrate and advance the system so that it could actually be using these trans well plates or 96 well plates so that you could just put your plate and assemble things simultaneously in all wells. So that kind of allows you to really assemble things after you step to be able to create them? Can you recover them? So we came up with different, as Dr. Mironov, these are like standard hydrogels that we use. We have, for instance, here fibrin, which is cross-linkable, chemically cross-linkable. We know it's about 10 minutes for the fibrin. It takes about 10 to 20 seconds to assemble things, and then we just wait for the fibrin to gel so that you assemble. These are the pink parts are the cells, so you can actually see a very high density of cells. This is really high that you would achieve with spheroids, and then you can actually get this tissue engineering. So high density of these cells right a minute after, at the beginning with the construct, that can allow early interactions and function real tissue constructs. So another important thing which I know that is highly valued in the field by experts is to be able to actually take matured spheroids and then assemble them. So we did also that, not only cells but also spheroids. So one thing that I want to make a note of here is we don't use any other materials to assemble the cells. It's just cells coming together or spheroids coming together. So we are not dependent on scaffolds. Only part that we use an external scaffold is to hold everything together, and it could be pretty much a biodegradable gel, but the scaffold doesn't come between the cells or between the spheroids for assembly. So we do our assembly, we now assemble spheroids, and then over time we watch them merge and form spheroids, integrate with each other. So we have also made co-cultures to evaluate this merger of spheroids after assembly. So these are fibroblasts after assembly. This is like a big circle, but we are looking at a smaller area on it. These are HUX spheroids that are already pre-stained with green. They are live, but they are green, so we can actually assemble them together and see the green HUX cells and the fibroblast spheroids and watch them over a period of 72 hours and see them merge completely. So that's an indication of being able to assemble these spheroids together, like Fred showed in the previous talk, and how we look at these spheroids merging. And I'm sure you will see Dr. Mironov talk about it in the coming presentation. So we have looked at some functional parameters. Now we have here not only a single cell type, but 3 cell types. We have fibroblasts, we have HUX, and we can actually track these cells to form these different gels. For instance, we see them secrete collagen type 1 and 4, which we didn't have earlier in the material. It was just fibrin. So we are looking at some of the formations, the HUX, that these cells are forming some extensions and flapodia, so to speak, in these constructs. And when you look at 2 spheroids over time and you start seeing them to form some connections, and this is how the merger takes place. And here the space is larger than we usually see. So this is kind of validating our assembly process of tissue spheroids in with multiple types of spheroids, of course, in 2D. Or we also started thinking about if we could control, again using acoustic waves in Z dimension, if we could make things multi-layer or multiple layers by again using acoustic resonances, not only in X, Y, but in Z. So there we built a system, and this one is not based on vibration, but it is actual piezo sending acoustic waves that travel to a reflector on the top and then bounce back. So now the standing waves are in the Z direction. This allows us to create nodes and anti-nodes, as many as we want, depending on the wavelength and the length. And you can that way assemble cells into multiple layers. And I'll show you more of these. And we started actually working on these with neural tissue engineering type applications. I won't go into too much into our engineering work, but the idea is that you have cells in suspension, so you have to first get them into suspension. You turn on the acoustics, and depending on the wavelength, you will have these multiple layers that are well separated. So we've done some simulations. These are published if you wanted to look at them. These are published, I think, last month in Advanced Materials. And you can see that the dependence of waves in Z and in X, you kind of create these plates. And we have some variation in the X direction also, because we are working in the near field acoustics, and it's always going to be different, but it still allows us to be able to assemble cells. We initially started with making layers of beads and controlling very precisely as a function of the frequency and wavelength where these layers will be. We have taken these embryonic stem cells and then assembled them here 1, 2, 3, 4, 5 layers. And this is like the construct kind of bends afterwards, and you can take these confocals. But the idea is you are pretty much generating almost a single layer of progenitor cells that you can then control the height between each layer. That was interesting to us because we want to see how neurites are extended between these layers, and if we could create multi-layer constructs where we can now in Z very well control the distance between the layers and differentiate these cells also in these multiple constructs and study their function. We're getting interested in their electrical properties, but what we want to really show is that we actually have a neural circuit, and that's mainly functionality. So the interesting technological part, or the powerful side of this technology, is you just, with the turn of a wave, you can very well start with well-controlled layers in a gel. In this case, we use either fibrin gel, and we have worked with GELMA as well, metacrystalline gelatin. But the bottom line is we have been able to take these cells and differentiate them over a period of 30 days, and these are biodegradable gels. Going above 40 days has proven to be challenging, but these were human cells, so we needed at least 6 months to really differentiate them to be functional neurons, and that's why we now have switched in the lab to, again, back to mice, which matures much faster, and working with primary cells to show that these actually are neural circuits. Let me show you one thing that's really happening right now that is interesting for us, that we are really pushing hard to prove, is the synaptic activity in these multi-layer systems. I think this should be probably the 30-day, I have to check back, but 1, 2, 3, 4, 5, 6 layers are taken here into picture, and you see how these layers that were flat layers of cells are now sending out these, wrong button, now sending out these neurites and neurite extensions. The red dots in this gel, and it's really hard to stain hydrogels in 3D, especially if you are looking for synaptic activity, but these red dots are, synapsin. So we are doing a lot of controlled experiments to move to, taking a lot of zoom-in images to look for these synaptic activity, synapsin stains right on the neurites, and to understand that if we have a system that is actually active, and it is hopefully at some point we'll be functionally able to prove that this is a neural circuit. Why is it important to show it's a neural circuit? Because you can really do this with iPS cells, which we have been getting really interested in. Then we could maybe start studying disease models such as Alzheimer's and so forth, and that's where we are really pushing this work. We have Thomas Neeland, who's a neurobiologist, who joined the lab a year ago from MIT Broad, and we are pulling together his neurobiology know-how and our tissue engineering, let's say, skills and tools to really move faster in the neurobiology, neural tissue engineering direction. So we are interested in levitation. We kind of like it. So over the years, I have been looking into magnetic levitation. If we could really assemble things, have them fly, and then come together in the and shape or designed for. So this is our 2011 work, the idea was not the most novel idea, I would say, but it turned out to be very useful and very interesting. What we did is we changed the material content of these gels and loaded their magnetic particles. And this made our gels and constructs these building blocks magnetic. So if we could control them and again pre-design what our magnetic field strengths would be. This is a simulation of a magnetic field distribution in a chamber like this, 1, 2, 3, 4, 5 magnets. And when you load your magnetically active paramagnetic ion-loaded gels, and they will just go ahead and align on these maximal magnetic areas. And this is like real-time what's happening. So that means if you can control what the magnetic field will be, which you can pretty much with solenoids or even like magnets that you can buy off the shelf or break your phone and you'll find a few in there. You can start doing interesting assembly systems, and you can start taking one type of a gel, control its magnetic strength, bring another one on top and bring another layer on top and go towards this direction. And now we've been thinking with the 3D biosystems how to maybe use this to make a system that would not only work on Earth, but potentially work on space. That's an interesting project that I'm looking forward to see how far we can go. But we are trying to just create a magnetized system and collect everything and have this done very controllably. One issue we had, not for pharmaceutical applications, I would say, but for tissue engineering applications to make an organ system or a patch, was that if you are going to implant it, since you have these magnetic nanoparticles, you see this is the meticylated gelatin SEM image, you don't have any of these magnetic nanoparticles in there. But as you load and make the gel more and more magnetic, you start seeing some issues with the porosity. And we had another follow-up paper where we looked at these biodegradable gels. As these gels biodegrade, the iron is being released and the cells are taking over the matrix. But still, for some of the applications maybe in humans, this wasn't the best idea. So NIH didn't like it that much, although I tried to really convince them 10 different ways with multiple papers. So we had to come up with a technology, let me go one step back, that you would be able to assemble things magnetically, but without magnetic particles. And one thing that we came across at the time was flying frog. And this is like $1 million system. It is liquid helium cooled and liquid nitrogen cooled. So it's like the size of this, I would say, this corner over here. It's very expensive. And it's kind of like the similar system that we use in MRI machines that are as strong as 12 Tesla. So since the distance is large, the gradient is very low here. And this frog is alive, by the way. No magnetic particles, no MRI contrast agents loaded to it. The frog has enough water in it that the magnetic susceptibility of water, when you have a large enough of a gradient, magnetic gradient, would be applied and levitated, beating or balancing with its gravity. So we took this idea, I think in 2013, and tried to change this system into a microfluidic system. So this is about 1 millimeter in size, you can just look at here, and this is just a capillary, like a microfluidic type system, where you have these single cells, and these cells are alive. We put them into an acidic environment, and the green is live, red is dead, and they are in this acidic environment starting to die, and the gadolinium that we use is going inside them, causing them to fall down. So we still create a similar, very strong gradient by not loading the cells with the magnetic particle, but changing and making the particles, the environment around it, paramagnetic. And that allows you to make a system pretty much that can cost $1 and can levitate single cells. And why is this important? I'll get into some of its applications and also show you what we are doing with it for spheroid creation or with microgel assembly with at the end. And it has very interesting applications in fundamentals of cell biology, cell density characteristics, and what it really means for cancer cells. But one thing that we realized early on is when you take this glass capillary, loaded with blood, you would end up seeing that the blood cells would form one layer, the white blood cells would form one layer, and there you might see cells like epithelial cells that shouldn't be there, most of the time being circulating cancer cells. And these are red cells in this 1 millimeter capillary that are being assembled, so they are going to the band where they deserve to be at, and this is just because that there is a magnetic force upwards and the gravity downwards. There is some, of course, Archimedes buoyancy force, but the bottom line is here you are looking at red cells with different densities. We took this blood from the same patient and processed it through a centrifuge to separate it into old and young cells, which are different in density, and we can kind of see that profile also here at a single cell level. Why is this important? It's important to apply this to circulating tumor cells, which people have been capturing with antibodies, epithelial type EpCAM-based antibodies. But if you look at kidney cancer, those cancer cells don't have EpCAM. So, and the capturing cells with magnetic beads and antibodies is very costly. That's why you could only process 7 to 10 mLs with the current technologies, and they're not that accurate. So what we are trying to do is if we could really pull out these cells by using some fundamental characteristics of cancer cells that are different, mainly density. So one thing that I'm trying to explain here is the heterogeneity in cancer, that all these cancer types have different levels of EpCAM expression, and as I said, some don't. And also, if you look at cancer, as the cancer cell becomes more metastatic, it goes through epithelial to mesenchymal transition, and there it loses its EpCAM or epithelial markers. So the most metastatic cells wouldn't have EpCAM, or you wouldn't be able to capture them with that antibody, which is the gold standard technology now. So what we do is, I'll just play this quickly. You've seen this video before, but this is like a real-time demonstration of how long it really takes to really stabilize cells in this system. And we are able to actually, within less than 20 minutes, create cell bands in a capillary format. What we learned, also investigating this further with cancer cells, blood cells, and these are mainly cancer cell lines. If you look at these different locations, these are all different cancer cell lines that we have looked at, and they have all different densities. And each dot here is the size of a cell versus a density of a single cell that you are looking at. And by the way, there is no technology out there, except maybe the AFM type technologies, that can measure accurately the density of a single cell. So here you're seeing the red blood cells, and that's where they kind of accumulate in terms of their density. These are white blood cells, and these are different cancer cell lines that we work with. So imagine that in patient samples, if you could draw a line between the blood cells, technologically, and the cancer cells, you would be able to separate circulating tumor cells without using any markers. So that has been our interest, and this is also another angle to it. Since that we can track over time a single cell's density, this is the video that you've seen, the cells falling down, you can see how resistant these cells are at a single cell level to a certain stimuli, such as a drug and so forth. And these are all actually published last July in PNAS and discussed in detail there. So we have then turned our attention in the past year to see if what we got to work with cancer cell lines would also work with actual cancer patient samples. We know in breast cancer that the cancer cells are epithelial, so they are larger and less dense, so they should be okay to separate. But in small cell lung cancer, the cancer cells are comparable in size to the white blood cells. And this is the blood that we have taken, we have gotten rid of the red cells, and we loaded it here, and we stained the white blood cell, and we put into the system also cytokeratin, and we realized that we don't only detect the single cancer cells, but we also detect these gatherings of cells, which are called circulating tumor emboli. These guys you cannot really get or capture with the current technologies because they are too large to pull. But in our system, since their densities are also the same as the others, you can pull them out. And another thing that's interesting that we are further investigating, and not conclusive fully yet, with animal models and human cancer patient blood samples, is looking at what these are. Are these activated neutrophils? Are they cancer cells that are not staining with cytokeratin? We used to always think, maybe 5 years back in the literature, that cancer cells had to stain positive for cytokeratin, positive for EpCAM, and negative for CD45, which is a white blood cell marker. But there is recent literature showing that that's not always the case. So we might be just seeing that here. So it's great to have worked with a microcapillary that takes about 50 microliters of blood. But my lab is at Stanford, is at the Early Cancer Detection Center. So if you are going to really detect cancer early, you have to really process a lot of blood. So for that, we had to turn this system into a high throughput system, and we have quite a bit of expertise in microfluidics. And what we ended up building was a system that would send the blood in, go through our magnetic gradient, and then have a wedge that would separate initially serum and white cells and red cells, and then if the cancer cells are above, they would be collected over here. So we kind of had this as an idea, and this is how we started, and this is, like, the system is quite small. Blood with the magic sauce comes in, goes through this system, and we have two outlets. Here, all you get in this is the serum, and you get all the blood cells over here without centrifuge, without doing any markers consistently, which could have very interesting applications, because if you wanted to get cells without activation, like neutrophils, centrifuging is necessarily not the best way to go, or even the beads. And we look at what we collect at the top channel, bottom channel. We look at the protein content, as well as, we shouldn't have any cells at the top channel. It should be all serum. We use a blood testing, automated system, and prove that, showing that, well, let me go back. If I can control where I put this separation, then I can separate things. And moving forward, we of course wanted to initially test this idea in beads that we know that they're of different densities. So these beads go into the system. They are color-coded, and they go out into 3 lanes. These ones that are actually blue, that appear green, and these are the blue ones, and these are the red ones. You see a mixture of the lowest density ones go in here, the medium density, well, the lowest density ones go to the toppest, the highest level channel. So you can actually separate different density beads. And it is becomes very accurate to tell that when you buy beads, they are not always the same density, the industry standard, because there isn't really a good way to measure bead densities. So what we have been doing now, moving this to cancer blood. These are blood samples, healthy patient blood samples, spiked with cancer cell lines first. And you're looking at the inlet of the system. The blood is flowing at 10 microliters per minute, 10 times diluted. And you see all the red cells here, a mixture of red and white cells in the second. And now you have to kind of, since it's not dark, be careful. You see, like, these are small cells that are kind of going into the upper chamber. There is another guy that's right going there. Then now you're going to see some emboli that are going into the top channel. If this is the video, I chose just one of the videos. But another guy just going up there. So these are spiked cancer cell lines into blood. Here, another guy that's, I'm sure you cannot see it from the far back, but the bottom line is, based on the density, we have a throughput system, a relatively fast system that can actually, in this big of a setup, separate cancer cells from blood without any markers, any beads, any antibodies. So, yeah, there is another guy going up there. So we kind of collect these on a filter afterwards, and then, do staining. So these cells are all alive during the process, and they can be done or taken to done further genomic and proteomic testing. So how do we take this magnetic levitation system and apply it to applications in biomanufacturing or biofabrication, in bioprinting? And that was where we actually came to, was by controlling the magnets and scaling this up. You kind of know when you put 2 magnets opposing each other, you have a point here that's the hot point that everything would be pushed to, that is not as magnetic as the environment. So we kind of take different gels which we have designed by playing with their material properties to be, well, before I go there, playing with their material properties to be different densities. And what happens is we put them in here, and you end up having these balance with the gravity force. So you end up seeing in this system, this is actually a large enough of a system where you see the different density gels going at different heights, like in the schematic that we are depicting here. And further, you can assemble these spheroids, or you can assemble small microgels in the same system. You just put them at the bottom, put it into the magnetically hot levitation area, and then these guys start going up and then go to that hot spot where they're all gathered together. And this takes, I would say, even faster, because we have very strong magnetic field here. These magnets are much bigger than these little ones that I've been showing you. So, and then these guys are forced together over time, and then they fuse together, cells become spheroids. And at the end, I want to show you where we are taking the system. We have just had a paper that got published on how we can integrate this system with cell phones. So you wouldn't need a microscope to do blood analysis or get white blood cell counts, or even monitor the presence of cells that are lighter density, which we believe today them to be circulating tumor cells, to maybe monitor relapse. And we built this system. So this is actually 2 magnets with a mirror on the side. The capillary goes in. This costs about $1 to make. And you just put the blood in the capillary with the solution, and then you just put it here. We kind of make it much larger to be able to do fluorescent imaging as well. But normally the system is very small. It's like a little tower. And then this is what happens. Oops, wow, that played really fast. Let me go one slide back. Go up. And then this is when you loaded the capillary. This is the cell phone image in 25 to 27 minutes. You kind of see the blood to come where it belongs. And you can actually build this system out of plastic pretty easily by designing these different parts and pulling them together. So it becomes a portable system. And you can even dilute the cells further, so that you would be able to not only see a band, but you would start seeing densities of cells loaded with some image recognition algorithms or programs in the cell phone. And these days the cell phone cameras are so good that you wouldn't really need a microscope with a small little lens. So this is where we are kind of asking now if we could take these systems and visualize how these kinds of new tools and technologies would fit in to the developed healthcare system. And these are a lot of questions in terms of where the point of care testing could be, or science could be in the future. And just to sum up, what I've been trying to show you is using these different technologies, or developing them, allows us to apply this across different areas, from even point of care to cancer to biomanufacturing and bioprinting. So these technologies are interesting enough and fundamental enough to cut across different disciplines. And I kind of thank my lab. They do all the hard work. We're about 13 postdocs and 17 students and technicians now in the lab, and this is in front of our center. I have to disclose this, that I have founded 2 companies due to the Harvard rules, which I'm still affiliated with. And I would like to thank you for inviting me here and for listening.
Thank you very much. Вопросы? Пожалуйста, не забывайте представляться.
Hello, Dmitry Sukharuchkin, Finance University. Did you tried to stimulate your neural tissue with a pulsed magnetic field or altering current?
So we haven't done that, but we have done some calcium activity testing by stimulating it chemically to see that the neurons are firing. But once, as I said, current goal is to show that it's actually something that is functional, that we can tell that neurons are communicating, and it's like a neural circuit. It's not just cells sitting there. Once we prove that exists, then it's a functional micro or mini tissue, and then all those things are possible to test.
Did it show SEG or AEG? Did you test?
Those are in progress. We are doing mainly right now chemical calcium activity, but if it shows actually calcium activity, then we know that there's also all sorts of synaptic activity there. So EEG and other things the electrical recording should be there.
Еще вопросы, коллеги?
Монавцов Игорь, к.м.н. Скажите, пожалуйста, вопрос по первой части вашего доклада. Можете ли вы утверждать, что волновой метод сборки клеток в структуры не вызывает повреждений клеточных или ядерных, что в последующем ведет к их ранней гибели или же наоборот к безудержному росту и превращению в рак? Спасибо.
Sure. So the first part where you assemble them with Faraday waves is not even it's not an active acoustic wave. It is just a vibration that you do. Oops. So it's acoustic wave. So you're just having the cells see a moving flow at the bottom. So that's really not affecting them. At the second phase, the waves that we are putting in, and I've worked in acoustic coupling to cells for a long time. When you're actually coupling acoustic waves to cells, what's important is the wavelength. So we are operating at 30-40 kilohertz. The speed of sound in water is 1500 meters per second. So at 30-40 hertz, the wavelength is at least 500, I would say, microns or more. So the cell size is 10 microns. So as far as the cell is concerned, there is no acoustic force gradient between the cell's two furthest edges. So the cell doesn't really see acoustic radiation force in that sense. So that would tear it apart. If you went to 1 megahertz, then that's where you do cleaning your petri dishes and so forth. That's a different story. So the range and the amplitude that we are at is pretty much the cell is surfing on a large wave on the ocean kind of thing. So it doesn't affect it functionally. One question that could come, I'm really trying to give a thorough answer to your question because I studied this quite a bit, is that if you are exposing cell to this mechanical effect, would it change its phenotypically, genomically, or proteomically? The answer to that is it takes for us about maybe 10 seconds to really apply the wave. So it is not long enough to create any shock proteins to really turn the cell into something that it isn't.
Hello, Anastasia Gorkun, Institute of General Pathology. Thank you for your impressive report.
Thank you.
Question about magnetic levitation of spheroids. Please say what type of cell you use. Did you use spheroids from different type of cells?
Yeah, so we have been working with iPS cells, human iPS cells that are differentiated into cardiac cells. So that's what we have been recently doing to control, relate the geometry to mechanical function. We have been working with human iPS cells differentiated into hepatocytes with some of my collaborators at Stanford to, again, implant them into animals and look at function. And in those, especially hepatocyte-type tissue engineering, we are not just using hepatocytes because to increase albumin and urein secretion, we have been adding or studying different ratios of stromal cells, fibroblasts and HUVECs, to understand if it has an effect on the functional outcome in terms of secretions. And we see positive effects if you have the right material and the right ratio. Right according to the what? It's a very interesting question.
Did you see, you show us that when you use, you can separate, yes, different type of cells. And when you use spheroids from different cells, you see this ring?
Yeah, you can actually. If you are going to magnetically levitate spheroids, I'll tell you something interesting that, for instance, if you take cancer cells you expose them to flow and shear. These cancer cells, we have shown in another paper a few years back, that go through epithelial to mesenchymal transition, so they become more metastatic phenotype. So their content changes. The densities of those cells, same cell line, that is exposed to shear versus not, their phenotype changes and their densities also change. That is within the same cell type. Think about different cell types.
Thank you very much.
Еще вопросы, коллеги?
Thank you very much.